Hi everyone. I hope you are doing great. In the fourth part of our discussion about the T cell activation, differentiation and memory, today we are going to discuss about the T cell subsets. Now these T cell subsets, they are the effector subsets or the effector cells which play a key role in our immune system to protect us or sometimes to create the disease. So let's see, what are the different type of T cell substrates and how they are contributing to our immunological consequences. Now, you know that T cell play a crucial role in immune surveillance and defense against infections and cancer. So we have been discussing this thing a lot, and we have TC cells and T helper cells. So basically, if we talk about the TC cells, they are the cytotoxic cells, while the T helper cells, they help to stimulate the immunology or to stimulate the immunological reactions by activating different type of cells, for example B cells, or for example the T cells, and by producing a number of cytokines which confer their effects on different type of cells. So subsets of both TC and TH cells, they have been identified and characterized. So we don't stop here just at the point of TC and TH cells, but there are other subsets also from these two different cells, and they play a very different roles, and they are they are generated by a different site of a different kind of cytokines. So what we are going to do today is to give you the picture about the TH and TC cell subsets. Okay, so let's start with the T helper cell subsets here. A naive CD4 positive T cell, it becomes a specific subtype by receiving signals from antigen presenting cells, for example, DCs, dendritic cells, cytokines which are present during the activation, and the nature of the pathogen. So when these three things, they signal a naive CD4 T cell, then it becomes a specific type of cell and it produces its subsets. Okay. Now each subset is characterized by polarizing cytokines. Now what are polarizing cytokines? Polarizing cytokines are the cytokines that initiate differentiation and that drive a particular T helper cells to convert into a particular subset. So these cytokines are called polarizing cytokines. Then we have the master regulator gene or the transcription factor. Okay. Now these master regulator genes or transcription factors, they controls the development of the subset. They are required. So first signal comes from the polarizing cytokines, and these polarizing cytokines, they activate the master regulator gene or certain transcription factors which controls the development of that particular subset. And then lastly comes the signature effector cytokines. And these are the cytokines which are produced by the fully differentiated T cells. Okay. So, see, the cell differentiation starts with the cytokine. So cytokines are driving the cells to differentiate into a specific subset, and then finally they are, once they are differentiated, then they are secreting their signature effector cytokines. That means again a set of cytokines which are unique or which are the signature cytokines on the basis of what these cells have been named. So let us see and let us understand what are the different type of subsets here. Now, this is a very clean example and a very clean picture about the T helper subsets and their functions. So now you can understand here. Start from the center. This is the naive CD4 positive T cell. Okay. Now this is the IL-12, IFN gamma and IL-2. They are the polarizing cytokines. So under the influence of these cytokines, the naive CD4 T cell, it activates a transcription factor called TBAT, and because of this particular transcription factor, the TH1 cell is differentiated. You got it? We start with the naive CD4 positive T cell. This is the polarizing cytokines which are driving the differentiation of the naive CD4 positive T cell into Th1 cell. And this in the box you will find the transcription factor or the master regulator gene. And once the subset is differentiated, then the function is, for example, in case of Th1, the clearance of intracellular pathogens like bacteria, viruses, or anti-tumor responses. Okay, so this is for Th1 cell. In the same manner, IL4 and IL2, they are the polarizing cytokines for Th2 differentiation, and the master gene controller or the transcription factor is GATA3. And then once these Th2 cells are differentiated, they clear the parasites, for example, helminthic worms. They play a role in parasitic immunology. Now, the third kind of cells are Th22 subsets, which are differentiated under the influence of IL-6 and IL-23, and the master regulator is AHR. And once they differentiate, they help in the clearance of the extracellular pathogens and epidermal immunity, that means on the skin. Okay, now T regulatory cells, they are are differentiated under the cytokines TGF beta and IL-2, and the master regulator is FOXP3, and they control the inflammation, the production of anti-inflammatory cytokines. Okay, now if you see the TFH cell, they are differentiated by the IL-6 and IL-21, which are the polarizing cytokines here, and these provide help to B cells in the promotion of the high affinity antibody production. All right, and then Th17 cells. These Th17 cells, they have the polarizing cytokines as TGF beta, IL-6, IL-1 beta, and IL-23, and they clear the extracellular bacteria, mucosal immunity, and other stuff here. And the last is the Th9 cells, which are differentiated under IL4, TGF beta, and IL2. And the master regulator is PU.1, and they clear the parasites and they provide the anti tumor responses. So, these are the Th cell subsets, their functions, and their polarizing cytokines in a single slide summary. Very useful for your examination. Okay, now moving forward, we also have the TC cell subsets, like T helper cell subsets. We also have the TC cell subsets, and if you can see here, the CD8 or TC cell subsets, they are also having the same name. Like TC1, instead of TH1 it is TC1, then instead of TH2 it is TC2, then instead of TH9 it is TC9, TC17, TC22, TFC, and then CD8 positive Treg cells. One is FOXP3 positive and another is QA1 restricted. Okay, two kinds are there. So these are the polarizing cytokines. So CD8 cell under influence of IL-12 and with the help of the master regulator T-BAT, EOMS and STAT4, it is converted into TC1, and then it secretes the signature cytokine IFN gamma. In the same manner, IL-4 influences the CD8 T cell to convert into TC2 through the master regulator GATA3 and STAT6, and the signature cytokines are IL-4, IL-5 and IL-13. In the same manner, CD8 cells, they are differentiated into TC9 by IL-4 and TGF beta with the help of the master regulator IRF4 and STAT6, and the signature cytokines are IL-9. Now, TC17s are differentiated under the influence of IL-6 and TGF beta with the master regulator ROR gamma T and STAT3, and the signature cytokines are IL-17A. In the same manner, IL-6 and TNF alpha, they help differentiation of CD8 positive cells to TC22 with the help of the master regulator AHR, STAT1, STAT3 and STAT5, and the signature cytokines are IL-22 and TNF alpha. In the same manner, here we have the TFC cells which are differentiated under the influence of IL-6, IL-21, IL-23 and TGF beta, and the master regulators are BCL6, TCF1, E2A and RUNX3, and the signature cytokines are IL-4, IL-21 and IFN gamma. And Tregs, there are two kinds. They are differentiated under the influence of TGF beta. So, the FOXP3 is the master regulator for FOXP3 positive C8 positive T regulatory cells, and EOMS is the master regulator for QA1 restricted CD8 positive T regulatory cells, with LY49 as the the protein, or the molecule expressed on the surface. So the FOXP3 positive regulatory cells, the signature cytokines are IL-10 and TGF beta, while in case of QA1 restricted CD8 positive Treg cells have perforin. Now you see, because their names, they are very much taken like or very much similar to the T helper cell subsets. So they closely resemble the subsets. Why? Because both the cells, the CD8 positive T cells and the CD4 positive T cells, they share similar signal 3 cytokine requirement. Now in my lecture about the 3 signal hypothesis, the signal 3 was the polarizing cytokine. So you can see here they have the similar signal 3 cytokine requirements there. Then lineage determining transcription factors, okay, they are also the shared one, and effector cytokine profiles which are secreted by them. And because of that, the CD8 positive T cell subsets, they closely resemble their CD4 positive T cell subset counterparts. All right. So now if we compare the CD4 T to CD8 T subset, so here you can see the polarizing cytokine for CD4 T cell is IL-12 for TH1. So here is IL-12 for TC1, IL-4 for TH2, IL-4 for TC2, IL-4 and TGF beta for TH9, IL-4 and TGF beta for TC9, IL-6 and TGF beta for TH17, IL-6 and TGF beta for TC17, IL-6 TNF alpha, IL-1 beta, IL-23 for TH22, but here it is IL-6 and TNF alpha which share where, and then they release the IL-22 and TNF alpha here. Now here IL-6, 12, 21, 23 and TGF beta, here it is 6, 21, 23 and TGF beta, and here you can see the same set of uh, the cytokines which are produced by them. And in case of T helper cells, the Tregs are just one kind of sets and they are under the influence of TGF beta, while in case of CD8 the two subsets are there which are differentiated by TGF beta. One is the FOXP3 positive and another is the QA1 restricted. So one secretes IL-10 and TGF beta while the other secretes the perforin. So that's a major difference between the T regulatory cells of T helper cell subset and T cytotoxic cell subset. Okay. Now let us quickly understand what are the different functions of the TC cell subsets. So TC1 cells, they produce perforin, granzyme, B, IFN gamma and TNF alpha, and their function is to eliminate tumor and infected cells. And the cytotoxic pathways which they take is the perforin, granzyme and FAS-L signaling. So this is basically the conventional role which has been known for very long, but recently ferroptosis and pyroptosis has also been included in their cytotoxic pathways. So additional pathways are there. Then in case of CT2, they stimulate IgE production, they recruit eosinophils and they contribute to allergic responses. In case of CT9 cells, they contribute into allergic asthma and atopic dermatitis. TC17 cells, they produce antimicrobial peptides and chemokines against the bacteria and fungi. TC22 cells, they show anti-tumor activity and protection against the HIV and certain other viruses. T follicular cytotoxic cells, they stimulate the B cells and they are the biomarker of the infection and cancer. And CD8 positive Tregs, they are basically immune suppressing cells and their role is to suppress the immune system. So this was, a very wide and very complicated topic which I tried to just consolidate for you so that you don't get confused, and in a few slides you get the whole idea about the TC cells and T helper cells. And this is something pretty very the I would say very exciting area where people are actively researching and they are just trying to figure out what can be the different roles or what can be the different functions of these T subsets and how we can utilize them for different therapies, for example immunotherapy for tumors and other allergies and some other stuff. So I will pause my discussion here for today's class, and thank you very much for watching this. Take care till we meet in the next class. Bye bye.